Tell us your name and about your project. My name is Dr. Rosalind Franklin. I worked as a physicist at the King's College London in early 1950s. My team studied DNA to figure out its 3D shape using X-rays. At this time, scientists believed DNA carried a genetic information, but nobody knew what it looked like. My most important result was photograph 51, an X-ray image that showed DNA as a double helix shape. Our goal was to take clear X-ray images of DNA and study the patterns. Those patterns helped us understand the structure of molecules. What were you trying to find? We were trying to discover the exact structure of DNA, how its parts arranged. Scientists knew DNA carried genes, but they didn't know its shape. We used X-rays on thin DNA fibers and studied the patterns they made. Photograph 51 showed an X shape, which told us DNA was a twisted, like a helix, like a twisted ladder. Where did the experiment take place? The work was done in a lab at the King's College in London between 1951 and 1953. We used special X-ray machines and spent months preparing samples. I worked long hours adjusting the setup and waiting for images to develop. Photograph 51 took us over 100 hours to capture. What else was happening in the world during this time? This was during the Cold War, when the US and Soviet Union were in conflict, and the Korean War ended in 1953. Nuclear weapons were also being developed. At this time, scientists in Cambridge were racing to solve the DNA structure, just like we were. Who else worked on this project? It was a team effort. John Randall led the lab, and Mr. Walkins also worked on DNA. I worked closely with my student, Raymond Gosling, who helped take photograph 51. Others on the team included Alex Stokes and Herbert Walson. Outside the lab, James Watson and Francis Kirk later used our data to build the DNA model. What was your role? I led the experiment. I prepared DNA samples and ran the X-ray experiments. I also analyzed the images and figured out what they meant. From photograph 51, I saw the pattern that showed DNA as a helix. By 1953, I had a strong evidence that DNA is a double helix, with the basis inside and the backbone on the outside. What impact did your work have? The impact was huge. It helped start model day molecular biology. It showed that physics could be used to understand life. It also helped scientists study protons, viruses, and genes. Our work made it possible to understand how genetic information is stored and used. What was the hardest part? The hardest part was preparing DNA samples. They had to be very thin and kept in perfect conditions. Even small changes could ruin the experiment. Sometimes weeks of work were lost in one experiment. Also, getting a clear X-ray image took a lot of patience. We didn't have modern computers to help analyze data. How do you hope your work is remembered? I hope people remember that careful experiments and good data are very important. Science is also a team effort. Everyone helps with something. Our work inspires future. I hope our work inspires future scientists to be patient and precise. How was your work environment? It was sometimes difficult. My relationship with Mr. Walkins was not always smooth, which made teamwork harder, but I stayed focused on the science. Do you think Roosevelt Franklin got enough credit? No, she did not receive enough credit for the work she done. Her work, especially in photograph 51, was very important, but it was also not fully appreciated until later in time. How do you feel about your discovery now? I feel proud, but also careful. The discovery of DNA helped medicine and science a lot, but it also raises ethical questions. I hope future scientists use this knowledge responsibly. Thank you. Thank you. It's important to remember these discoveries and continue learning from them. Thank you. Bye-bye.